Embryo. It is a process of development of zygote into an embryo. The process is called embryogenesis. So in embryogenesis, zygote forms embryo. Now this is a micropylar end, and this is the chalazal end, and this is the embryo sac. Development of embryo takes place at the micropylar end of embryo sac. When certain amount of endosperm is formed, the time it triggers the growth of embryo. Means growth of embryo triggers only after certain amount of endosperm is formed. Here zygote divides to form two-celled pro-embryo. So this is zygote, it divides to form two-celled pro-embryo. The two cells are unequal in size. The larger cell towards the micropylar end is called basal or suspensor initial cell. And at the chalazal end, the smaller cell is called embryonal initial cell or terminal cell. So this is suspensor initial cell, that is basal cell, and this is embryonal initial cell, that is terminal cell. In both the cells, cell division takes place at the same time, but for understanding, let's study this separately. So here, suspensor cell divides transversely in one plane to produce filamentous suspensor of 6 to 10 cells. So this filamentous suspensor is made up of 6 to 10 cells. The first cell of the suspensor towards the micropylar end becomes swollen and functions as a haustorium. The lowermost cell of suspensor is known as hypophysis. So this is haustorium and this is hypophysis. Haustorium is the first cell of suspensor. Hypophysis is the lowermost cell of suspensor. Suspensor helps in pushing the embryo deep inside the endosperm. Now let's focus on embryonal initial cell. It undergoes 3 successive mitotic division to form octant. And the planes of division are at right angle to each other. So here first it forms 2-cell structure, then 4-cell structure, and then 8-cell structure, that is octant. Now in this octant, the lower tier of 4 cells of octant give rise to hypocotyle and radical. So lower tier of 4 cells of octant, they forms hypocotyle and radical, whereas 4 cells of upper tier forms pumil and 1 or 2 cotyledons, depending whether it is monocot or dicot seed. This is hypophysis, which by further division give rise to the part of radical and root cap. Subsequently, the cells in the upper tier of octant divides in several planes so as to become heart shape, which then forms 2 lateral cotyledons and a terminal pumil. Further enlargement of hypocotyle and cotyledon results in a curvature of embryo and it appears horseshoe shaped. The embryo development is similar in both dicot and monocot up to the octant stage. The differences appears later. In monocot embryo, single cotyledon occupies terminal position and pumil is lateral. The protective sheet of pumil is called coleoptile and that of radical is coleorhiza. Finally, ovule is transformed into the seed and ovary into the fruit.